Welcome to the Microscopists, a bite-sized bio podcast, hosted by Peter O'Toole, sponsored by Zeiss Microscopy. Today on the Microscopists.
Welcome to this episode of the Microscopists, and I'm joined by W.E. Moerner of Stanford University, and we discuss interdisciplinarity.
You know, I'm hard to put in a box. That's certainly I'm a card-carrying chemist.
Discovering jumping molecules.
So these jumping molecules, that was a truly exciting time because that was a total surprise.
The importance of music in W.E.'s life.
Singing Gilbert and Sullivan, and that connection is actually how I met my wife.
And being a procrastinator.
My thesis, the papers that came out of my thesis work did take several years after I left Cornell to actually get them published.
All in this episode of the Microscopists. Hi, welcome to this episode of the Microscopists. I'm Peter O'Toole from the University of York, and today I'm joined by W.E. Moerner from Stanford at UC San Diego. How are you today?
I'm wonderful. Thank you very much. Great to see you, Pete.
Yeah, and thank you for taking the time to join me today. I've got to say, I'm going to start slightly off tack. I'm looking behind you and I can see a ton of books. So are they all factual or are they fictional or a mix?
These are really all factual. They are covering sort of the many fields that I've been involved in. Elsewhere, we have some fictional books or things like that, or historical fiction books in the house. So this is my home collection of these books. There's a similar collection in my office at work.
Actually, do you know, this is going in a different direction to normal. I believe this background picture I just put in Zoom, if you're listening, this is W.E. in his office at work. And one of my quick fire questions is often, are you a messy or a tidy person?
So I would say that this particular picture, if you look closely, you actually will see little Post-its along the edge of the bookcase, right along the lower edge of the books. Okay. Yep. And those Post-its actually provide the dividers and the sections in which the books are organized. So it turns out to be very organized in a certain way, and in the sense that I can find things pretty well in this office. So that's the sort of aspect of this sort of, if you like, cluttery mess that makes it a little bit redeeming for me.
So it's an organized mess. It's not a mess, not a tidy. It's not a mess. It's an organized. Organized, I don't know what, I'm better be careful what I say, but it's quite good actually having the virtual background, having your library at home, merging into your library pretty much at work, and just how many books there are. It is an insane amount. So when you said you had some fictional books elsewhere, what sort of fiction are you into?
Well, actually historical fiction, books like The Swerve, I've enjoyed very much because it's talking about those early days and the monks that preserved sort of the early history of the Greeks and so on through the Middle Ages, things like that. I enjoy that the most.
Okay. So that was a complete digression to how we usually start. So I guess from my perspective, you're most well-known, most famous for super resolution work and your Nobel Prize in chemistry back in 2014. When you got that Nobel Prize for the recognition of your tremendous work before that, did it make a change to how you perceived work after than before?
Yeah, it's a really good question. There's some ways in which there were changes and some ways in which there weren't. I actually continued and want to continue and have continued doing research with my students and postdocs and so on, because I enjoy working with them. I enjoy the challenges of the new things that haven't been done and all of that. But of course, the Nobel Prize made a huge change in other aspects of my work, just because of a bunch of sort of side effects, if you like. There's people who overly revere the Nobel laureates, and therefore invite them to come to meetings and so forth just to have a Nobel laureate show up. They don't care what you say. and that kind of thing. So there's been a big mixture of trying to balance those sorts of things with my desire to sort of continue science.
Okay, so what got you into science? Let's go back. What got you into science to start with? In fact, no, do you know what? Let's go back a bit further. When you were the age of 10, around that age, what did you want to be when you grew up?
Well, at that time, I was very excited about lots of things technical. Because my father had been trained in electronics in the service. And so I had some electronics books to read from him. Also, he was a chemist when his degree was in chemistry. So I had a chemistry set in the backyard in a metal building that I called the shed or something like that. Or the playhouse or the clubhouse. That's it, the clubhouse. And I could go out there and, do chemistry experiments or do electronics experiments. So I was just at a time just sort of soaking up science and really enjoying all aspects of mathematics and all of that as well. So this, I think, came mostly from genes, but also from the times, which depended upon the world sort of reacting to Sputnik and things like that as a driver in the United States to encourage young people to go into science. So my parents were encouraging me strongly to go into science or engineering or something like that.
So your Nobel Prize is in chemistry, but would you ever class yourself as a chemist?
I'm hard to put in a box. Certainly, I'm a card-carrying chemist. Okay, I've been a member of the American Chemical Society for years, decades. I teach general chemistry to students and so on. But in fact, my career has had an evolution, moving through many fields of science, starting with engineering, adding physics and mathematics, and all of my primary degrees are actually in physics and so on. And then in my career in industry, there was a lot of physical chemistry added, and which I enjoyed tremendously. And then much more chemistry, and now biology and so on. So I'm really a person who enjoys all of these.
Do you have a favorite? What do you have a favorite area of science, though? If you could just concentrate on just one area, or do you love that multidisciplinary approach to it?
Well, that's an interesting question. What I think I enjoy most is methods. What I enjoy most is applying engineering, physics, and chemistry to make something happen, something new happen, and to analyze, to calculate what's the best signal. How can you detect a single molecule? I mean, that was sort of the big driver at the beginning, how to detect that single molecule, which is a combination of several of these fields. And I still enjoy that because every time we improve our detection, then we measure something more. And the applications to biology right now, of course, are the most interesting to me because there's such a world of problems that need to be explored, all of which have heterogeneity and all of which have different behaviors that can be observed a little better if you observe single molecules one by one.
So it's interesting that all that multidisciplinary, the ability, that flexibility to do quite broad research. I think academia is fairly unique in the ability to enable that freedom. I did actually do some background research, which I don't always do, but I noticed that you worked for IBM for a while.
Yeah. Yes.
Did that have the same freedom to operate?
Well, I actually think that's the opposite. If we go through what you just said a little bit and think more about it, academia has historically been dominated by what people often call pillars of excellence. So there'll be a professor who doesn't collaborate with anyone and has a huge crew of people working around him and doesn't learn a lot about what's happening across the street or in the other buildings and so on. That's kind of the way it's been historically. Now, this, of course, has changed a lot recently. There's a lot more multidisciplinary work in many universities now. The comparison to the corporate research laboratory is an interesting one because I was at IBM Research and one of those great industrial research laboratories of that time where you could pursue basic research, but and rather than just applications to products. Okay, and so that Bell Laboratories is another example, and there's more examples that of that type, but they've gotten smaller or changed in more recent years because there's been more competition pushing companies more toward just worrying about trying to get the products out. In any case, the aspect of this experience that relates to your question is we often were there trying to work on a big problem. Let's say optical storage. I was heavily involved with optical storage, and this was going to be by any of multiple techniques, not just pits in a CD, but actually writing bits into molecules absorptions, okay, at low temperatures, something called spectral hole burning. And this spectral hole burning idea was intensely focused on the properties of molecules absorbing light. And so I had a lot of opportunity to study that, but we collaborated with engineers who wanted to figure out, will this ever work? Will it be practical? What will be the cost for all of this? And with chemists who were making the molecules that enabled this hole burning storage. So physicists, chemists, engineers were actually at IBM Research all working together. So there was very few barriers between departments because of this focus on getting the project to move forward and learning whatever you needed to do to push the project forward. So it's a fascinating thing. And I'd sort of view myself as bringing that kind of viewpoint, to academia, as one of the people who brings it to academia.
And as you rightly say, that mindset of that pillar of excellence, one group being very insular and being very big within themselves is, I think, hopefully broken down almost, not completely. I'm still aware there's these singular pillars within different institutes around the world, but mostly most big groups are team players now. And I think
that's true.
Really important.
But there's still people that say, are you a chemist or a physicist? And I have to say both. And we're doing biology and using engineering. So, there are some people who kind of push this pretty far.
Well, I think there's an advantage. Once the pillar becomes team playing, it can associate, it can work with chemists, physicists, biologists to really optimize and get the best out of science and speed science up.
Right.
I think is important as well.
Yes. Absolutely true.
So that must have changed a lot. You say you brought that culture in, or helped certainly develop that culture. Did you meet much resistance from certain groups?
Well, a little bit. I would say that some people historically might have said, maybe we can't hire this person because he's not really a chemist, let's say. And others have to explain, how can this person fit in a chemistry department and so forth. So that battle was won, of course. But, it might not have been won 10 years ago, but 10 years before I arrived.
Yeah. So on a completely different note, do you remember your first microscope?
Do I remember my first microscope? Well, if you allow me, there certainly have to have been some microscopes in high school when we looked at samples and looked at a frog dissection or whatever, because we take a biology course, we take a chemistry course, we take a physics course.
Yeah.
And so that it is certainly there, but very dim in the memory because it's hard to remember things that are that far back. A more relevant comment about that is it's important to remember that all of my entry into this whole field started with spectroscopy. So the variable that you change is more frequency or wavelength. And that's an axis on which you plot things, okay, as opposed to a spatial axis. So, which is more commonly the way a microscope is viewed as having spatial axes, okay, X and Y and Z or whatever. And so the microscope that's worth mentioning, perhaps in terms of sort of a more important microscope was when we were looking at single molecules at low temperatures at IBM Research, okay? We started making hybrid images that had frequency and space. So it was essentially a confocal microscope because it was one position, but you at a time until you move it across the sample. And then we also were changing the other variable, okay, the frequency axis, and that can turn into a two-dimensional plot. That's a hybrid plot. Okay, so that's a pretty important microscope because it was really one of the first times that we scanned over the shape of the single molecule spot, which is really the point spread function of the system.
So exciting times. And yeah, so I guess the first research grade microscope at that point. I actually have in my office my first microscopy lesson at university that wrote down. Actually, I don't ask me how, I just opened a book that I was thrown away, my university notes, and it was there. I just opened it on that page. I thought,
great.
I've got to take this out and just put it on the wall and just realize how bad I was at microscopy back then. Or even how much I hated microscopy back then. The quality of the microscopes were not that great. And then obviously the world's got a lot better and microscopes have got a lot better. So you sent some other pictures. So this is obviously dating back somewhat. So this is definitely your chemistry side of things. Let's describe who we've got here and what you're looking at.
Well, that's actually my physics PhD. Okay. There's a Professor Al Sievers, okay, with the beard, and Andrew Kreplevy, an older graduate student, a year older than me, who was a collaborator on my research work. And what I'm holding, if you can see it down at the bottom, is a model that shows the perrhenate ion, the ReO4 minus ion, the perrhenate ion, which we had doped into a crystal of an alkali halide, such as potassium iodide. So the potassium iodide structure is shown there. Some of the atoms of the potassium iodide structure is shown with a tetrahedral ReO4 molecule in the middle of it. So it's a really interesting sort of juxtaposition that you bring up here, because that research, you might call it at the time chemical physics, because it was a molecule being studied by physicists. There actually are some physicists, even as famous as Art Shallow, who said if a system has more than two atoms, then it's chemistry, so it's not physics. But here we were physicists studying this perrhenate ion and its infrared properties, its infrared vibrational mode absorptions in that solid, which was like a host holding that perrhenate ion. All of this at low temperatures. So that's sort of an interesting juxtaposition of physics and chemistry and so forth, helped me in sort of my transition toward more chemistry, if you like. But we were very interested in the properties of how that behaved at low temperatures, how it would change when you irradiate it with light, how it would rotate inside the crystal, change to a different configuration, make what we call the spectral hole, and those kinds of optically induced transformations, you see, have permeated my career.
Yeah, what was really great is your passion talking about that. It seemed to bring back actually quite a lot of excitement. So, what's been the most fun time of your career?
I would say the most fun time was when we were beginning to study these single molecules at IBM with my postdoc, Pat Ambrose. And the experiment was looking at the pinosine molecule in a crystal of paraterphenyl. I mean, that was the first system where we could see a single molecule. And one afternoon, by the way, at the research laboratory, there's the research room, the laboratory, and then right across the hall is my office. So I could go into the laboratory or in and out or whatever anytime I needed. But one day, so you have to imagine what's going on. As we're scanning the frequency of the laser, then this was so-called excitation spectroscopy. When the frequency of the laser reached the resonance frequency of the molecule, the optical absorption frequency of the molecule, That's at 500 terahertz, by the way, or 593 nanometers. Okay. The fluorescence would increase, and then you would detect more signal. And then as you tune further, it goes down again. So this is well known, as you might expect, but this is in the frequency domain, very much like tuning your radio, far away from any city, and you can mostly noise. And then finally, when you get just to the right frequency, you hear the signal from a particular station. So that's the molecule, coming into resonance and giving us fluorescence. Okay, great. So we were studying those line widths and all this business, right? But Pat, one afternoon, came running into my office and he said, the molecules are jumping around. They're moving from one frequency to another. They're moving around. And this was really exciting to us because we're at 1.2 degrees Kelvin, okay, in a crystal. We didn't really expect to see a lot of big dynamics, but the molecules would actually, their resonance frequency would move from one frequency to another, to another, to another, to another, back, and so on. So this turned out to be, the beginnings of blinking, okay, that molecules would be in resonance or out of resonance, and their local environment will be changed by the optical excitation, which would then cause their resonance frequency to shift. So the optical excitation was changing their local environment, molecules would shift back and forth. So these jumping molecules, that was a truly exciting time because that was a total surprise. It was one of these things that turned out to be, well, might have said that you could predict it. I mean, the theorists had for a long time been saying that molecules, can move around in frequency space in glasses, in systems that have sort of more loose dynamics, let's call it, because lines would broaden. Well, these spectral holes would broaden with time, which was a suggestion that the molecules were moving around. But here we saw these digital jumps from one frequency to another, and in the crystal, which really surprised us. That was very exciting.
So, and I very good explanation of it. Did you realize at the time exactly what that meant when you saw that they were jumping about? Well,
we sort of quickly thought that, well, maybe this is spectral diffusion. Maybe this is a direct observation of spectral diffusion that sort of had been described by theorists previously for glasses. But having it happen in sort of a discontinuous way was a wonderful surprise. And then the grist of theorists who then studied this in more detail, like Jim Skinner later on.
So from all that, all the research, obviously I like this picture because it is, you describe, this is your Nobel, right? Also I presume your wife and your son.
That's right. That's my wonderful wife, Sharon, my son, Daniel. And we're holding the medal, the Nobel medal. But the other beautiful thing is the so-called diploma. And that painting there is an original painting drawn just for my prize. Each Nobel Prize has a diploma like this associated with it. So this was, of course, a great moment after the award ceremony in Stockholm.
And so obviously your wife and son must have been very proud. What do you do in your spare time with them? I presume you have spare time.
Yes. Well, since the prize, there's actually been less spare time. But one of the spare time things that we spent a lot of time on historically was music, singing and music. And so I actually met my wife through having fun with singing. Our son had a lot of fun with singing in high school as well. And it turns out that singing was something that you may recognize more in the UK. It was Gilbert and Sullivan, singing Gilbert and Sullivan. So singing Gilbert and Sullivan, and that connection is actually how I met my wife. So very nice sort of connection occurred through there. She comes from a family of very talented people and singers and performers. Her her father and mother were the director and producer and partial founder of the Gilbert and Sullivan Society of San Jose. And so they put on, over the years, 20 to 40 or whatever productions, and several operettas were done every year. And so I picked this up actually in college because I had a very close friend, Burr Stewart, who got me involved with listening to Gilbert and Sullivan. Along with playing instruments, clavichords, and he would play the flute, I'd play the clavichord, and things like that. And eventually, I got involved in Gilbert and Sullivan, listening to it a lot in college, and then in graduate school when I'm supposed to be getting a PhD. In the middle of that, I decided I'm going to go try out for the lead of HMS Pinafore. Out of the blue. And that was something that I was encouraged to do by a wonderful postdoc, Don Trotter, who said, "You got to try, go ahead and try out," right? And so forth. So I went there and I sang for, the captain, and I got the role of Sir Joseph Porter, Knight Commander of the Bath, KCB. And so in grad school, I performed that role, which was great fun, but of course it takes too much time. To do that, I had to stop after just one show, get back to working on my PhD. But that is why when I moved out here to IBM in California, I easily decided to go try out for roles at the Gilbert and Sullivan Society of San Jose, where I met my wife and so on. And that's the story after that.
And this is a picture of your wedding?
Yeah, that's right. Yeah. You really pulled out some good ones. Then? Sorry?
How young were you when you married?
I was 30. Okay.
I'm glad you got that right. Otherwise, you'd have been in trouble when your wife listens to this back. That would have been just dreadful at that point. Yeah. You're testing my memory, okay? As I get older and older, memory, those specific sort of things. But so you see in that picture on next to me are her father and mother, the Gilbert and Sullivan experts. Of course, they're both physicians and had stunning careers. And then my father and stepmother beyond that. And then on the other side of my wife, Sharon, is her sister, and brother. And then my very close friend Burr is the very tall person with his wife, Barbara. So, it's quite a family picture here. But the amazing thing about the Stein family, that's my wife's family, is that all of them would get involved in these Gilbert and Sullivan shows. So all of them would perform in some way or another, or be involved with the lights, or be involved with the stage, or be involved with being a producer, or be involved with, and costumes and building sets and so on. So it was a great fun activity. So I was going to ask you if you started anything, but obviously you did get roles. You described that quite nicely. You have had roles in that. Do you still do it?
So what happened is, in terms of music, being in a big show requires a lot of rehearsals, and then requires full-time rehearsals, the week before the performances start, so-called Hell Week. I just wasn't able to dedicate that much time, given all of my research commitments and so on. So I shifted more to singing with large singing groups. They mostly sing classical works, and a San Jose Symphonic Choir, Schola Cantorum. So all these different singing groups I've been a member of over the years. The one that I was involved in for a good number of years was the Stanford Symphonic Choir. which would sing a lot of large oratorios and things like that, which I enjoyed tremendously. But since the prize, I haven't had time to do that. The thing about the voice that's particularly nice is that you don't have to rehearse a lot. You do have to go to rehearsals, but you don't have to sort of beat on it every day like I had to do with, let's say, when I played the bassoon in high school, or when I played the clarinet, or those sorts of instruments. You really have to practice and practice and practice, all the time. But singing is more natural for me, and you carry the instrument around with you and so on. So I'll get back to it as soon as I possibly can.
So thinking about getting back to things on a more serious note, when over your career do you think have you found most challenging during your career?
One of the challenges that I found to be something that I spent a lot of time thinking about was when I decided to change careers, when I decided to make the big transition from IBM to academia. This was difficult at the time because IBM research was a place where you sort of stayed there for life, historically. And one of the things that was going on during the early 90s was this large change in the computer industry. The emergence of the personal computer changed the landscape tremendously and affected IBM. So there was one year where there was $8 billion in losses at IBM. And this sort of changed the research division from a place where there were a large number of people working on basic science or important things for the company to fewer people working on basic science. So I felt the environment changing. I felt my opportunities sort of reducing somewhat. We had detected single molecules, which was tremendous excitement for me scientifically and for all the people working in that area. But IBM actually wasn't so interested in that. They were more interested in STM and moving atoms around and things like that on the surface. And so I had to make this choice to switch to the academic environment with its exciting aspects, but also challenges. You have to wear many hats in academia. You have to get grants. You have to write proposals. And you have to, of course, mentor many graduate students and postdocs, and be on committees and teach and all of that stuff all together, all at the same time. So it's actually been really exciting and a challenge. But I enjoyed the challenge. I mean, the challenge was something that I think broadened me, and of course the contact with many young people in academia has been spectacular, and the freedom was higher in academia. The freedom to explore whatever science I can get funded, so I could then switch to look at biological systems, which I wasn't able really to do at IBM.
So go on. So now you're at this point of your career. If you could research anything, forget the history, forget what you've majored in, what would you go on to do? What would you like to see researching? What would you like to succeed in?
So, the current trends in science, many of them move toward quantum effects. And the power of quantum mechanics and its ability to provide special forms of computing and so on. Historically, that's been studied mostly by physicists, again, on small systems now, on atoms, some number of systems that involve defects in solids. And there's not as much with molecules. And there's such a power of molecules in the sense that there's so many different structures that can be built and can be imagined and so on and so on. And we've actually shown, that molecules have power, can be used for, imaging beyond the diffraction limit and all sorts of things. And so having new systems available where there can be sort of real entanglement in quantum systems and molecules for quantum mechanical applications, I think that would be exciting to be able to do that.
What's stopping you?
One interesting thing about all of this field is having the right materials. Having the electronic states and the interactions with the nearby environment and so forth that maintain the wonderful properties. Another way of saying that is molecules are highly quantum mechanical already, but typically for very, very short time scales before what's called dephasing or decoherence sets in, and decoherence destroys these quantum effects. So one has to come up with schemes to get around that.
So it's interesting. So obviously one of your fellow Nobel laureates at the same time was Eric Betzig. And I remember Eric, and I asked about what he wanted to be as a child, and he said an astronaut. And I asked him what he wanted to be when he got into science. He said an astronaut, but he's not. And asked him what he would be today if he could be, and he'd still be an astronaut. But actually, you also have a passion for space.
Right.
Some wonderful images of you outside I guess a giant signal detecting telescope. Where are you exactly in this picture?
So I am at what's called the shack of the Stanford Amateur Radio Club up on the hills behind the university. And this is the radio telescope, one of the radio telescopes. This one is only about, let's say, 10 meters in diameter. It's not the huge radio telescope. There's one of those also up there. But this is for the ham radio operators to use. So this one is one built and maintained by amateur radio operators. And so this is another one of my hobbies. The beautiful aspect of that kind of a dish is that it's powerful enough to transmit a signal toward the moon and bounce it off the moon, so that signal can reflect from the moon and come back and be detected again by the same antenna sometime later after the propagation time to the moon. This is so-called Earth-Moon-Earth transmissions. And so this is one of the fun activities that the amateur radio operators get involved in. I'm not being irradiated. I'm off to the side of this, of the antenna a little bit in this picture. But that night, we couldn't see the moon because of those clouds that you see out there. Nevertheless, we could bounce signals off of the moon. So one of the really fun things to do. So amateur radio and the connection with radio, the connection with electronics and frequency domain, and all of that is throughout this particular hobby, which has become very digital now, but it's still using all of these wavelengths and so forth that make amateur radio work.
It's a lovely interconnection, and I have to show it because it's such a wonderful image. So this is Hale Bop. I think I remember Hale Bop. A comment. So is this your own image that you've taken?
Yes, this is my own image, and there's a great story behind it. So this I believe the year was 1997. Yeah, 1997. We are at that time I was at UC San Diego, University of California at San Diego, in La Jolla, California, down in Southern California. That's where my first faculty position was. And that was the faculty who took the chance on this crazy guy, from IBM. And the event there is a special one because there was also a lunar eclipse at the same night. And so my wife and son and I headed out to the Anza Borrego Desert. So this is from the Anza Borrego Desert, which is inland from the coastal cities down in Southern California, down San Diego area. And I was just having a ball because I had one camera aimed to the ocean. That's the direction to the west of where the Hale-Bopp comet is, over the mountains there. And the other camera was headed to the east because that's where the lunar eclipse was occurring at this moment. So to do this, we pulled our trailer out to the desert and set up for camping and so on. And our son was 6 years old at the time, so They came out with me in the dark to see this crazy thing going on, but they quickly got tired of sitting and watching and taking pictures and so on in the dark, so that they went back in and to go to the camp, and I stayed out there taking these pictures. And it was really, really great, because after a while I started hearing sort of a bunch of coyotes, making a huge racket, very nearby, eating something. Okay, so I quickly grabbed all of my cameras and headed back in to the campsite, but got these great pictures at the time. A bit of a sad note, because this is the same night that this crazy group of people decided that there were aliens behind the comet, and so on. And so this is the people that drank the Kool-Aid that particular night, unfortunately.
Wow. Keeping on a happier note, it's fascinating the interlink of yourself and Eric, both into astrophysics, astronomy. And we've got, I don't know if in the UK, we've got a program called Sky at Night. I don't know if you've ever come across, it's probably one of the oldest still running TV programs. Mm-hmm. And that's presented by Chris Lintop now. And actually, he's been really important now with the image analysis of biological images, especially in the 3D electron microscopy community.
Oh, fantastic.
It's those connections, spectral and mixing. So Scott Fraser with all the spectral. Again, it came from the astronomers and what they were using, bringing it into the life sciences and into the microscopy world. There's fantastic crossovers. Which also, again, the multidisciplinary approach, the working with different partners, and sometimes the importance of blue sky research, because you never know where it will lead. And
yes, that's a really very, very important point. And I didn't mention it earlier, but I would like to go back to that great day when we first saw that spectral diffusion. That was a great example of this. Okay, people say, well, why do you want to try to detect a single molecule? They were sort of making fun of me at that time. What good will it be? Who cares? So we don't want to know what one molecule is doing. We care about the average value. And of course, the real reason for doing it is that you learn new things. You're looking in a regime where you haven't looked before, and you may not know everything that you're going to see. And that's exactly, what that example illustrates, and the many other examples since. So this is why a blue sky researcher pushing back boundaries, or looking in places where we haven't looked before, and things like that, all still have great value. And I'd like to connect with what you just said. You mentioned electron microscopy, and you mentioned some of those things. Some of the newest things that we've been doing in my lab, and there's a great postdoc, Pete Dahlberg, that's been working with me, and he and I have built this system where we do both single molecule imaging with fluorescence and electron microscopy on the same sample, precisely the same sample. So this has the effect of annotating the electron microscope image with the single molecule positions. See, annotation has been done for some time, but that's by people sort of identifying a membrane and then coloring it in, or identifying ribosomes and coloring it in. But there's thousands of proteins that you can't identify with your eyes. You can't see, you can't recognize them. You don't know which protein is which. So there's thousands of hidden proteins in an electron microscope image. So with this single molecule experiment, we light them up, and we can say where they are to within, the precision we get from the photons detected on the order of, let's say, 10 nanometers, which is far, far better than doing that from the diffraction limited regime, and providing information of, let's say, where in a cell, what is the cellular location of a particular set of proteins this way.
Yeah, after this, we'll have a quick chat about that in more detail and see where that fits in. We've done some work with Lucy Collinson in the past, which was driving down a very similar route. So it'd be interesting to cross some notes on that. Some quick fire questions. Mac or PC?
PC.
PC. Early bird or night owl?
Early bird.
Early bird. Do you have any bad habits?
Being a procrastinator.
Oh, you've done well to publish so much if that's your bad habit. How do you publish so much if that's your bad habit?
It's been a fight. It's been a battle. My thesis, the papers that came out of my thesis work did take several years after I left Cornell to actually get them published. But my wife helped a lot in sort of getting me to finish those papers. And she's actually helped me cure me of the procrastination business. But still, okay, if there's an interesting, I'll give you an example. This last weekend, there was a reenactment of the 1921 first transcontinental radio signal, radio message being sent across the Atlantic. Okay, so this is a big deal. It's the 100th year anniversary. So the hams in the United States decided to reenact, and so they were going to send from the East Coast and so on. So I had to go out there and set up my radio and try to see if I could also detect it. Of course, they're sending the signal the other way. They're not sending it west. I didn't have really a good antenna. It didn't quite work, but it was fun. and that was something that kept me from, writing the papers or the letters or whatever that I was supposed to be doing this weekend. So anyway, there's still procrastination.
So I think there's a lot of PhDs, postdocs who are very reluctant to publish their work because there's always one more experiment that can be done. So how do you draw the line and say, no, we've got, you're a procrastinator. So how do you actually say, no, we do have to go with this now? Do you have any advice for those who struggle to get to that point and actually commit to publishing?
Well, I guess I'd like to make a distinction between being a procrastinator and being a perfectionist, because I think they're a little bit different. Procrastinator is doing something else rather than working on what you're supposed to be working on. A perfectionist keeps thinking, I got to do it a little more perfect, a little more perfect, a little more perfect. Okay, and that's something that also can slow down publication. The answer to that question is a tricky one. It depends upon the experiment. It depends upon the system. It depends a lot upon assessing. What have we learned now? What have we learned so far? Have we learned a number of new things? And if we've got a reasonable number of new things, then there's a great value in sending it out to the community to so that other people can learn about this and start working on it as well. And I'm more in that category than taking a super long time before trying to submit something. I have to admit, though, let me just say there's always good and bad sometimes on certain things. Part of this is because our culture and research in the United States is more tuned to getting results, publishing papers, and getting more grants. And because of the challenge of getting grants, because of the incredible challenge, there were many years that I did not get grants on our new biological imaging work. You know that you better publish in order to get the crucial funds that are required to support the group. As a university researcher, if you don't bring that money in, you can't pay the students. So that's a very harsh realization, that makes academia different from what was going on at IBM. And so that, I think, unfortunately, also pushes us sometimes to publish a little faster.
And a lot of pressure at work. And I presume, is this a lot of your lab?
Yes, you can see we're under great pressure there in that wonderful moment.
Well, I can see them all wearing hats of some sort, which is always a very sign.
So if you can't quite see it because of your shoulder, but down on the floor, if you can move slightly more, maybe you can see the tip of a bottle of champagne. So there is a bottle of champagne down there on the floor. And the student in the middle, Josh Yoon, did graduate here. Okay, this was the result of a successful, either a paper, published in a high-level journal, or a successful thesis defense. That's the kind of thing that we have these celebrations for. And he's wearing a protective helmet. Okay, because we're going to shoot the cork down the hallway as far as possible. So the goal here is to shoot the cork down the hall of the laboratory as far as possible. And you see, there's other interesting hats and helmets and so on. I have one on, but you may not be able to see that. But everybody is there, cheering on the group member who has recently achieved something important, and for this sort of ceremonial shooting of the champagne cork. So this is just, I sent you this picture because it's kind of a wonderful example of some of the crazy things we do to have fun. We don't just work. We have some fun as well with things like that. And in that picture, I want to show you something else. If you show it one more time, if you can.
Of course.
You may notice that there is a belt bag around my waist here. Okay, so that is not quite understood by many people, but I find it a convenient way to have the phone and the wallet and even a flashlight and a few important things that you might need in an emergency. It's all coming from ham radio and emergency communications kind of stuff. You want to be ready for what's going on. So as part of a farewell party, the students made some ceremonial belt bags. With this one says no ensemble averaging on it. That designed by Allison Squires, the woman's down here with the maroon cap, and the other student, whichever students were also leaving at the same, postdocs leaving at the same time, as a ceremonial item.
Do you wear it?
Oh, this one, I'm still wear the old ugly looking black one. This one's so important. I couldn't possibly, I might get it old or dirty. I have to put it in a special place inside the house.
That's why you always need two of everything, so you can actually enjoy wearing one and keep the other one for keepsakes.
That's right.
So back to the questions. And actually, my next one, you're going to, I know the answer already. TV or book? I think I have to say a mixture. Okay.
We watch the news quite a lot on TV because we end up looking at the news while exercising, let's say, in the mornings. And books, of course, are something that I actually would like to read more. I'm unhappy that I haven't read that many books, and I would like to read more. It requires turning off all the other lists of things that have to be done. And to dedicate time to that.
Okay. What's your favorite film? Your favorite movie?
Contact.
Okay. And I wish I'd asked every guest this. I've only asked a few. What's your favorite?
Although I have to say that the new Foundation series is really spectacular. Okay. Isaac Asimov, many episodes, 10 episodes of the Foundation has been really, really great. But I don't think it displaces Contact.
What about your favorite Christmas movie?
I have no idea. I'll just say that this week I happened to get some crazy enjoyment out of watching the woman who puts on this annual Christmas show about The Only Thing I Want for Christmas Is You. Mariah Carey's Christmas show was just nuts and this year and fairly entertaining because of all the crazy things that were done.
That sounds like good fun. So, okay. Tea or coffee?
I'm more of a coffee person, but I mix in teas also. So it's like half and half these days.
Okay. Are they short coffees or are they big coffees?
Big coffees. More often we do a brewed coffee rather than espresso.
Okay. Beer or wine?
Wine.
Red or white?
Red.
Okay. If you were to be taking out food, what's your favorite food type?
I think I would say seafood. I think I would say, salmon. We have lovely salmon out here, so it's really wonderful.
And do you have any food hates? If you're dished up, so you go to a conference, you're dished up something, you feel, I really don't like that. What's your nightmare food?
I don't know. I'm really omnivorous right now. didn't used to like collard greens, let's say, because my parents fed it to me a lot. I guess that might be something I don't enjoy as much.
And who cooks at home?
Well, it's more, it's my wife, although I cook my breakfast every day. We, these days, since we're cooking a lot at home, are utilizing one of these services that sends fresh food to us, and then we have to cut it all up and so forth and cook it. And so she often does a lot of the cutting. Sometimes I do a lot of the part of the frying and so on, or not really always frying, but cooking, you know, and finishing off some of the dishes. So we tend to share when I can.
Okay, now we are nearly up to the hour. Incredibly, it goes way too fast. I'd like to take you back, actually, and I have read your Nobel piece, and you actually sent me this picture of your parents.
Mm-hmm.
Do you think they've been the biggest influence on your career, and who else has been an influence on your career?
Well, they've been a tremendous influence in the early, incredibly formative days up until age 18 or so, you know, after finishing high school and heading off to college. During that time, it was that oh so important time. My mother was reading to me very early on. My father gave me a lot of appreciation for the sciences and through his connection with physics, chemistry, and electronics. And he was also, by the way, in terms of images, he was a professional photographer. That was actually his career. So a lot of images there and so on, and chemicals because of the developing. But anyway, they just gave me a tremendous support in my work. And even though most people, or there's a lot of people that don't want to leave Texas, where I grew up, they still supported me when I went far away to Washington University in St. Louis, rather than going to one of the universities in Texas, which are also very good. It was better for me to see the bigger world in terms of the overall development, and they supported it anyway. So, you know, I was an only child, so it was a tough thing for them when I left. Of course, the other major influences are mentors that have been really important along the way. Professor Jim Miller at Washington University was the one who got me more involved with experimental physics and projects in the laboratory, in his laboratories at Washington University. And he had a tremendous also personal influence in terms of advising me how to navigate through all the complexities of college. And then the other really important mentor is my thesis advisor, Al Sievers, that you saw in that picture with the molecules. So, you know, I've profited from a number of really, really good mentors, even at IBM. Gary Bjorklund at IBM was a key influencer in FM spectroscopy, which is what I use to detect a single molecule. So there's a tremendous crew of people that I've been very thankful for.
And I also note that actually you gained a scholarship very early on, which you actually paid tribute to being a significant part, because none of this would have been possible without a scholarship in your particular case. Right.
That was the influence of a very important counselor at the high school. So at the high school, there were many, many things going on. I was involved with On the Spot. I was in the band. I was in speech and debate. I was in all kinds of events. But of course the courses in science as well. But the counselor let me know about this very special Langsdorff Fellowship competition at Washington University, and that was a key influence because she urged me to apply for it, and I eventually got it. And being able to go to Washington University was a key step. Yes, absolutely right.
And on one final note, because we are up to the hour, I can't help but admire your tie that you are wearing. Which actually, I'm not sure. It's not a big screen. What exactly is it?
Yeah. So if you look at more of it, you see that we got many colors of the spectrum and so forth. This is the SPIE tie, one of these societies that provides conferences on imaging microscopy, lasers, photonics, and all kinds of things. And has a tie like that. I thought it might be kind of appropriate for this today.
Now I'm embarrassed not to be wearing my Royal Microscopical Society tie, but I'm not a big tie man myself. I'm very much an open collar. So
I'm not normally either, but this is a special occasion to be able to talk to you like this, Pete.
Yeah. So WE, thank you very much for joining me today. Everyone who's watched or listened, it is actually worth a quick watch as well to see some of the images that WE actually sent in. But WE, thank you so much for your time today. It's been great to meet you, get to know you personally, and I hope everyone's really enjoyed it. And don't forget to subscribe to the channels. WE, thank you very much.
It was my pleasure. It's my pleasure. Thank you as well. I really enjoyed chatting with you.
Thank you.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.